In this lab, we will explore the morphology, or the different shapes and structures that we can find on our prokaryotes, which will be bacteria. And the way that we're going to accomplish the completion of this lab is using several websites that have very good micrographs or microscopic images of the different structures that we're going to observe. The first image that we have here is a monotrichous arrangement of flagella. So this corresponds to the first image that you need to draw. And you see a lot of different bacterial cells here, but I want you to focus on these here that are a little bit larger, a little bit darker, that you can see. So what you're seeing with these particular specimens is a rod-shaped bacterium with a single flagellum flowing out from it. Mono means one, trichous pertaining to hair, and that's how we or that's the term that we use for flagella. So a monotrichous arrangement would be one single flagellum on the end of the bacterial cell. So this would be a monotrichous arrangement where we have this bacterial cell with one single flagellum flowing out from behind it. Our next arrangement that we have is what we refer to as lophotrichous. Lopho means tuft. So in a lophotrichous arrangement, we have a tuft of flagella that appear at one or both ends. In this particular example, we have lophotrichous flagella at both ends of this spiral-shaped bacterium here. So you can see what looks like these little wispy hairs, and you can kind of understand why we use that Latin root for hair, that trich, T-R-I-C-H, to represent the flagella, because they do look like little hairs. So this would be a lophotrichous arrangement. This arrangement that we see here of this particular image is what we refer to as amphitrichous. Amphi means both, and amphitrichous in this instance means one single flagellum at both ends. The image that we just saw of the lophotrichous would also be an example of amphitrichous. So amphitrichous can refer to a single flagellum at both ends, or a tuft, multiple flagella at both ends, just as long as we see something at both poles or at both ends of the bacterium. So amphi means both. Our last flagellar arrangement that we have is referred to as peritrichous. And this is where we have flagella that go over the entire surface. So instead of these being polar, instead of these being at the ends, you see they're all over the surface. So we have this rod-shaped bacterium, and here we actually have two that are kind of still stuck together. And you notice all of the different flagella coming out from it. So the flagella would be covering the entire surface of the bacterium. Now, when we refer to the shapes of bacteria, there are three main shapes that we see. We see the cauci, the bacilli, and the spiral. The cauci, or coccus for singular, refers to circular or round-shaped bacterial cells. This figure that you see here shows the different arrangements that we can have of the cauci. Because they are round, spherical, they can divide in multiple planes. So we see more arrangements with the cauci than we do from any of the other bacterial shapes. So we can have a Diplococcus, Diplo referring to two, so that there are two kind of still stuck together. We can have Streptococcus, which is a chain, almost looks like a chain of pearls. If the cauci divide in two different planes, we can end up with a tetrad, which would be four cells kind of all stuck together. If they divide in three planes, we can end up with a sarcina, which would be eight cells stuck together. And then if they just divide in random planes, kind of in all directions, we can end up with what's referred to as the staphylococcus or staphylococci, which would be a cluster or kind of a random grouping of the cells. So we're going to take a closer look at these and draw some of these actually as they would appear, not just in an illustration here, but what they would look like if we were to take samples of these bacteria and stain them and then look at them using the microscope. In this particular image we have here, this is Neisseria gonorrhea, which is the causative agent of gonorrhea, and it tends to exist in nature as a Diplococcus arrangement. So where you see these arrows here, we have this Diplococcus arrangement. But you'll also notice that we have little areas where we also see just single cells. So you can use this one image here for two different drawings on your lab document. Number 5, which is the coccus, which is just the singular round cell. And then number 8, which is your Diplococcus, which is the paired arrangement that we see here. So one of the kind of characteristics, if you will, of these Diplococci is that they exist in nature just like this. They'll be stuck together due to how they replicate. So this is helpful for diagnostic purposes because if we took a sample, for instance, from a female vagina that was suspected of having gonorrhea, then we could do a gram stain and look for that Diplococcus arrangement that could give us a definitive diagnosis for gonorrhea. So these different shapes and arrangements that we see can help us in diagnosing different infections. So we have the singular, the coccus, the one singular cell, and then we have the Diplococcus where they are still stuck together. Our second shape that we look at with regards to bacteria is the rod or the Bacillus shape. And so that's what you see here. We have this smear. This is actually of E. coli. We have this smear of, 100 or so rod-shaped cells. Now, the thing that you have to keep in mind about Bacillus is if you see Bacillus or Bacilli written, non-capitalized, not in italics, we're referring to the shape. We have a genus, an actual scientific group of rod-shaped bacteria that go by the name Bacillus. For instance, Bacillus anthracis. That's the scientific name of the causative agent of anthrax. So we can use the term Bacillus to refer to the shape or to refer to the actual genus name. So just make sure that you pay attention to that. So this is our rod-shaped, our Bacillus bacterium. There are 3 different types of what we consider spiral bacteria. And the first one of those is kind of a mix or kind of the linkage between a spiral and a bacillus, and that's the Vibrio or the curved rod. These kind of look like commas. So where you see these arrows here, you've got these little comma-shaped bacteria. So these are referred to as Vibrio. Now, Vibrio is another one of those that we have to be careful with because Vibrio can be used to describe the shape or it can be used to describe the genus. Vibrio cholerae is the causative agent of cholera, and so you have to be careful again using this term just like with Bacillus. So the Vibrio is the curved rod, kind of the link between the rod-shaped Bacillus and our other spiral structures that we'll see. Now on lab 3, there is only one circle for drawing your spiral bacteria, but what I would like for you to do is kind of divide that in half because we're actually going to look at two different types of spirals. Our first one we have here is the spirillum, and this kind of looks like a little spring here or like a little S. They can be a little bit more coiled, but we tend to kind of see them in these little flexible kind of curves here, kind of resemble S's. So this would be an example of a spirillum, which is one type of spiral bacterium. Our second type of spiral bacterium is called a spirochete. And you'll notice that we don't see that S shape or that really cute little spiral shape that we see with the spirillum. These are a little bit different and they tend to behave differently. The spirochetes tend to almost corkscrew or spiral their way into tissues or through the skin. And so the spirochetes are going to take on a little bit different appearance than you noticed from the spirillum bacteria. So just use that one circle to draw both your spirillum and your spirochete. Another structure that we can look at as it pertains to bacteria is the bacterial capsule. So bacteria are surrounded by a cell wall, but some types of bacteria have this extra layer, this extra coating surrounding them called a capsule. And what you see in this image are some rod-shaped bacteria, okay? And then you see kind of this negative space around them, this space that isn't stained, and that's the capsule. So what we've done in this image is we've stained the bacteria, we've stained the background, and now you can see where the capsules are. Not all bacteria have capsules. Not all bacteria of even the same species have capsules. And in fact, later on in the semester, when we talk about, what makes one bacterium able to cause disease and another not, one thing that we're going to consider is the capsule. So the capsule can make certain bacteria become pathogenic. The last bacterial structure that we are going to look at here is what we refer to as the endospore or the terminal endospore. So what we've got here is Clostridium tetani, which is the causative agent of tetanus, and one of the best examples of an endospore former. So you've got these rod-shaped bacteria here, okay? And you'll notice on some of them, the cell itself appears fairly light color, and then you've got this darker structure kind of at the end. Makes it look maybe like a lollipop. I always think of those like safety pops that used to get, like for Halloween when you were a kid, or even a tennis racket. And what you're looking at here is the terminal endospore. Certain bacteria can form these endospores that help protect them when conditions aren't very favorable. So in this particular example, in the Clostridium genus, they're notorious spore formers. They can survive for years. We're not even really sure how long they can survive in their spore form. But when we stain them, we can see the vegetative cells here, but then we can also pick up these endospores in staining.